Thanks so much for giving me this opportunity talk. Trying to decide if this is theory. It's definitely not mathematics, and it might be more just speculation. But I don't know. Maybe that's part of this. So I work on transposable elements and how they're regulated. And there's two classes that are worthwhile knowing about. Class 1 retroelements, which encode, well, there's the gene there, and then they encode an RNA, and then the RNA encodes for a reverse transcriptase, and then the reverse transcriptase through cis preference stays attached to the mRNA that it was its source and goes back into the nucleus and makes a copy, so they replicate that way. And then the DNA transposons, actually, in some ways, they're more clever because, well, they make a transposase, it excises, it goes somewhere else, and then well, they can jump ahead of the replication fork the way that Manus talked about, but sometimes if they leave a break on a sister chromatid, then they'll repair off of that and they'll replace it. Sort of, they hijack the DNA repair machinery to kind of amplify as well. So there's an alternate mode of DNA transposon replication. They're mostly harmful. They insert into genes. That's problematic. They can also, because of their repetitive nature, if a double-stranded break occurs in one of them, if there's non-allelic recombination, you can cause big chromosomal aberration. And different species have different mutation rates caused by transposons. So in fruit flies, about half of all visible mutations that are caused and been found for past 100 years caused by transposons. In mice, about 10% caused by IAP elements. Humans, it's much less than that. It's interesting to think about why there's variation in that. Different transposon families can vary in the degree to which there's a sort of embedded nature within the genome. The line 1 element, that's the most famous one, most famous transposon, I would say, in humans. It's been resident in mammals for more than 160 million years. So all these mammals outside of the monotremes have these. Some they invaded probably in the theory of there, but other types of transposable elements are kind of like flash in the pan. So the P element transposon, that just invaded probably about 100 years ago in Drosophila melanogaster. So the sort of duration of which these things remain and retain in these genomes, I think, is an interesting thing to think about, the degree to which they may have co-evolved with their hosts. But despite the fact that they're harmful, there's a lot of interest now in the ways in which transposable elements can provide the fuel for adaptation. So it's sort of like a double-edged sword here. So here's the adaptation world. Maybe this is a fitness landscape. I don't know. That's a very sad fitness landscape on the other side. But the adaptation, I think, can be fueled through what are called direct modes. That's kind of more understood. I'm going to do a quick walk through what I would call the direct modes, but then there's also the indirect modes, and I'll talk about, I think that's a little more interesting. We actually heard a little bit about potential direct adaptation when Manisha gave the talk this morning in maize. Transposable elements can provide really interesting fuel for cis-regulatory evolution. So here's a gene, and in blue, I'll show these transposable element fragments. They could be DNA transposons or retrotransposons. The thing is that the transposable elements, TEs, they have to contain sequences that recruit RNA polymerase, host RNA polymerase, or splicing factors. And then they also have to do things like regulate their expression either in the germline or maybe to prevent expression in the soma. So there are regulatory functions that are kind of within the context of the way that the host regulates genes. So that's an interesting thing. Here's a great example of a retrotransposon it's an endogenous retrovirus-like transposon that actually has been shown to be important in regulating this gene CDK2AP1, which regulates the cell division. In mice, well, historically, there was this other transposable element that led for exon 1 skipping in this gene, and then this skipping allows a new isoform to be expressed that's important in gestation. And actually, this has been supplanted with another transposable element insertion in the mouse lineage. And now this one actually is the one that's taken over the regulation of this isoform. And if you delete that transposable element, this guy, you see that you upset embryonic growth. So that's regulatory function. But they also provide functional sequences like protein functional sequences. So Ts encode for proteins of all different kinds. They have to bind DNA, like the transposases have to actually recognize DNA sequences. So there's this DNA binding function, and they actually have to manipulate DNA sequences, and they also enable cell-to-cell transmission for things like endogenous retroviruses. So these proteins can actually contribute to protein function, and probably the two coolest examples for that are with regard to VDJ recombination. So to generate antibodies, you have to shuffle the V domain, the D domain, and the J domain. And this is the RAG1 nuclease that's shuffling and doing VDJ recombination to generate antibody diversity. But this protein actually evolved from a transposase. This is actually a transib DNA transposon. It's still active in some type in lamprey. And then in embryogenesis and gestation, the placenta, there's this really intimate connection between the maternal side and the fetal side. And again, actually, there was a phylogeny that showed this syncytin proteins again and again and again through mammalian systems. Retrovirus-like proteins have adopted an important function in mediating the interaction in the syncytial parts between the placenta and the mother, and that's evolved again and again and again from endogenous retroviruses. And so this is a third direct mode of adaptation that you can see. This is actually when I call this genome maintenance through true domestication. Oftentimes people will refer to modes 1 and 2 as a form of domestication. And I don't know, I have a little bit of a pedantic point I want to make. But the point about this is actually it's the continued transposition of the transposable element lineage has been captured by the host to maintain some sort of maintenance of genome function. And so my pedantic footnote is that, you know, if you have a lineage that's been, well, it's still, these can all continue to reproduce. And I would say that those are examples of domestication. But when it's just like a piece, like wood, or like an antler that's being used for a coat hook, or like baleen maybe used for a corset, I don't know if those are good examples of domestication where you take a chunk of something and then you just repurpose it. I feel like it kind of has to continue living for it to be domesticated. So that's just me ranting and raving a little bit. But the best example of this, what I would call domestication, is at the Drosophila telomeres, because instead of, to solve the end duplication and replication problem, there's a retrotransposon array at the end of the telomere here, and the transposons have evolved the ability to go back into the telomere. So the telomere shortens, and then they get reactivated, and they start jumping back into the telomere, and you can maintain the telomere in that way. So, you know, there's a lot of examples, like more and more examples. The more we sequence genomes, the more examples we can find. So I think it's interesting to think about what makes transposable elements such an effective source of adaptation. And I think it's because they're really high information content mutations, right? A SNP, well, there's not a lot of information when a G goes to a C, but when a whole chunk of something lands somewhere, there's a lot of information in that form of mutation. But what I also think is interesting is that information that exists within the transposable element got imbued within the transposable lineage through evolution on a fitness landscape, which is in the host genome itself. So I kind of think of these transposons, especially the ones that are long residents of these lineages, they're kind of, adapting on a fitness landscape in the host, right? They have to use the host machinery to move around. And so they may end up at places, sort of coincident places on a fitness landscape, where the information that is now embedded in the transposable element is not simply functional for the TE lineage, but then can also be functional for the host. So it's almost like this information from one adaptive landscape is moving to another lineage when they insert into a place and it's beneficial at that location with this particular function. So those are direct modes, and then indirect modes. This is actually something that I'm really interested in because I study the way the transposable elements are regulated. So they're harmful, right? They cause mutations, they cause genome instability, and in response to that, there are a lot of different mechanisms of evolved genome defense to suppress the activity. And I think that that's an important part of what has happened with regard to adaptation, because those evolved systems of genome defense can also be recruited for gene regulation. All right, so I'm going to provide some of my favorite examples with regard to piRNAs, because I study these Peewee interacting RNAs. piRNAs are like an immune system that fight against transposons. So here's a transposon mRNA, and then here's a piRNA, Peewee interacting RNA. This is a Peewee protein, and they recognize, actually, they're like little antisense nucleotides. They're 23 to 30 nucleotides. So that ensures specificity. And then the Peewee protein cleaves the transposon mRNA. They actually they don't just cleave. They can do things like go into the nucleus and silence things through chromatin. So piRNAs are in animals are like a really important way in which transposable elements are regulated. So here's their suppression. But piRNAs again and again and again and again and again seem to have been recruited for gene regulation. So in C. elegans, the 21UX1 piRNA downregulates exolethal to ensure robust dosage compensation. In Bombyx mori, the silkworm, there's a piRNA on the W chromosome that regulates the mass expression to ensure sex determination and proper female development. And more recently, people have figured out that if you knock out certain piRNA clusters in mice, you can render mice infertile, because those piRNAs aren't regulating transposable elements. They're regulating genes that are important for spermatogenesis. So there's this, the walk through this is like transposons, you evolve the piRNA machinery, and now this piRNA machinery has this capacity to expand its domain and its function beyond transposon control into the role of gene regulation. So that's kind of convenient. Actually, more broadly, it's interesting to think about. This isn't some new idea on my end, I want to be clear about. But there seem to be a lot of candidates for genome defense mechanisms that involve secondary function in gene regulation. Actually, or maybe RNA silencing in general. But you do need to say is like, well, what's the historical function of RNA silencing? Is it virus control? Is it transposon control? Is it gene regulation? It's a little hard to say. Maybe the ancestral function is transposon control. I'm biased, I would say that. Or maybe DNA methylation is another system of genome defense, and chromatin-based silencing. This is a cool paper. I like this quote. This is a recent paper looking at polycomb. Polycomb is thought to be an important developmental regulator, but here they say the conserved polycomb repressive complex prevents transcription of protein-coding genes in eukaryotes. In a broader level of eukaryotes, its preference is towards transposons over genes, suggesting that repressing transposons is an ancestral function for the polycomb response machinery. You can even go beyond gene regulation. Actually, just biotech. RNA interference and CRISPR and restriction enzymes. These are genome defense mechanisms, but they're also useful like to cure people, right? Because they can turn genes off that are harmful. So I would like to finally say that, like I said, like, okay, well, we have these direct modes, and then we have these indirect modes. Maybe the TEs have evolved put pressure on the host to evolve a silencing machinery that then can be secondarily recruited. But actually, I think these are quite enmeshed, because a lot of these things I just talked about, like the transposable elements that are at the telomere, or the cis regulatory sequences, or different splicing factors, or functional sequences, or things that regulate mRNA stability. These are all TE fragments that mediate these different functions, but the way in which they mediate these different functions actually seems to depend oftentimes on the host regulatory machinery itself. So all of these different things, PEG10 is an imprinted gene that's regulated by chromatin silencing, and there's a lot of other interesting things. The Mellon sex determination is a transposable element insertion, but its way in which it mediates sex determination depends on DNA methylation. So I'll be quick here. Maybe I'm overtime or undertime. I have no idea. It doesn't matter. Because I'm done no matter what. So I'd like to sort of read a couple takeaways. So internal conflict Ts can provide raw material for hosts directly via these high information content transposable elements insertions. The information content within these variants is partly put into the TEs by their evolution on the fitness landscape by evolving in a host. Now I say partly because there's a lot of horizontal transfer too that transposable elements do. So maybe it's a fitness landscape over here. They evolved on that fitness landscape. Then they moved into a new, and maybe there's some correlations between fitness landscapes, or at least the structure. But an indirect mode of adaptation is driven by T's, the evolution of these silencing mechanisms that are then later repurposed for gene regulation. And I think actually it's really difficult to tease these two things apart, because these host silencing factors actually may have facilitated that original sort of direct mode of adaptation. So I think these things kind of work all in concert together, and that's it.
Maybe time for one or two questions.
Yeah, do one question, sure.
Oh wait, I got the microphone.
Oh yeah.
The voice is loud, but yeah.
So the impression I get about, the impression I'm getting from this workshop is that these TEs are so numerous in so many genomes, right?
Yeah.
And then there's this whole sophisticated network of repressor mechanisms. But are there good examples of kind of the thing going on in real time? So the invasion of new TEs spreading across genomes, and with mechanisms of suppression that aren't quite there yet? Or is it all mainly historical, what we study?
Well, I think actually earlier when Manisha spoke, we saw that earlier step this morning, actually, right? So we see the sort of and all the ways in which she was showing that the TEs might be regulating gene expression is an initial step, because those are polymorphic within the population, right? And so maybe some of those are adaptive and maybe some are not. So then there will be a winnowing step, probably. Now, have we specifically seen examples whereby in real time we've seen adaptation triggered by a transposable element that swept through a population? There are examples like that. For example, like the what's the name of the moth that's in the UK? Huh? The peppered moth, right? So there's an example of adaptation because the pigmentation changed in the moth in response to industrialization. So there is an example there. Now, whether or not that depended on the host silencing machinery interacting with the transposable element itself, I don't know off the top of my head, but I think it did act through splicing. So yeah, so I think that's probably a good example of kind of inaction. Yeah. All right, thank you very much.